now I'll talk about another project that we did, and a huge credit to this goes to so many people, especially Dr. Lakesh Sharma's team, because we were trying to collect some additional data from on farms, which was actually correlating to the small plot research that Dr. Lakesh Sharma and his team has been conducting at NFREC Swanee Valley for the last couple of years. So I'll share what we have done, or why we needed to do these, and I'll be open for your discussions and suggestions. So I'll start with why are we doing corn nitrogen rate trial? The first thing is that our current UF recommendations are outdated. They were based on research conducted several decades ago. And now with advent of new genetic material, with new precision ag techniques, I guess, we were due for a new updated rate. And along with, as Jay was mentioning, that most of our farming falls under basin. Our growers are basin growers, so we fall under BMAPs. And so there's environmental impacts, regulatory inspections. So, our growers are under, pressure to basically comply with the current UF IFAS rate. And that rate, as I mentioned, is outdated. So I think we are in the right time frame-wise. We are in the right time to basically have a study and update our corn nitrogen recommendations. And Dr. Lakesh Sharma and his team is working diligently on that. So before I move forward, I'd like to just give a brief history of our corn advisory committee. So small plot research trials began back in 2021 at NFREC Swanee Valley. At that time, Dee Broughton used to be our regional specialized agent in row crops. She formed a corn advisory committee that would guide and assist with the research trials. And that committee consisted of corn growers, crop consultants, IFAS extension and research faculty, and NFREC farm manager. So since the inception of this committee has guided these trials by providing several vital suggestions and helped in developing protocols. And then out of their suggestions came on, it was their suggestion to have on-farm trials. And we'll talk about that, in the coming slides. So this is a slide that I stole from one of our presentations that Dr. Lakesh Sharma and his postdoc, Dr. Diego Litao, they shared with the Corn Advisory Committee meeting in January. So this is just for everybody to know that how much engaged our Corn Advisory Committee is with these corn nitrogen rate trials. And on your left-hand side are the different nitrogen rates, pounds per acre. So this is how the trial was initially set up, from all the way from 0 to 350 pounds with a 70-pound increment. But if you look across from left to right, for each rate, how much nitrogen split will be applied was basically discussed and approved by the Corn Advisory Committee. So all these numbers, all the way from this 70 rate to 350 rate, when nitrogen application needs to be applied and how much it needs to be applied, was discussed and approved by the Corn Advisory Committee. So that is the extent of their input into the protocol development. And I'm not going to share all the protocols, but this was just for you all to see their involvement. And before I move forward, I just want to show you guys that over the last several years, we have invited the Corn Advisory Committee members during the growing season to, at different growth stages, to come and have a look and they have shown tremendous amount of interest. They have visited one-on-one sometimes. They have also visited in a group of 2 or 4. They have also visited in a big group to see the rate trial, to discuss what's going on, and to suggest us what we could do better. So, but they have played a huge role, in moving this project forward. Now, as I said that, every year when Dr. Lakesh Sharma's team collects data, analyzes it, and by the time, we have our annual Corn Advisory Committee meeting, we present that data to them. And based on what they're seeing, they might suggest us to change something or to update a protocol. And that's how the corn on-farm trials came. But I just wanted to show you, and these are the next couple of slides, or I guess 3 slides are again from the corn nitrogen rate trial. And as you can see, on the top half, the 2 graphs are from site 1. So these small plot research is conducted on 2 different sites at Swanee Valley Research Station. And the bottom 2 graphs are for site 2. And if we are looking at these data points on individual years, you see there is a lot of variation, and these are replicated 4 times. As now when Dr. Sharma's team did the statistical analysis, there was no year effect, so both the years were pooled together. And now you can see that the variation, the error bars are now lower. So this is I just wanted to iterate that now we are just talking about a 8-row, 30-foot long plot replicated 4 times, and we are seeing huge variations, right? And I'm trying to make a point here. So when we, did the same trial next year, we didn't see a year effect, so the variation became a little lower. Now, when we didn't see a site effect, now you can see the variations even became lower, right? So this is basically, this is how the first couple of years worth of data is looking for the corn rate trial. These are not any way, shape, or form the recommendations, because we are still undergoing, Dr. Sharma's team is still undergoing the trial. But at the same time, the question is, if we are seeing that variation in small plots, which all those replications are situated within a couple of acres, and we see those huge variations, now, the point that I want to make is that the nitrogen use efficiency, small scale versus large scale will not be the same, because when we are looking at a small plot, but when we look at a field under the pivot that is 160-acre pivot or 120-acre pivot, that field might have a lot of variations in the soil due to soil properties or due to management differences in the previous years. So whatever we are doing, or whatever the data that we are getting from small plots, may not correlate or correspond to what our nitrogen use efficiency will be, or what our response will be when we are actually growing corn on large scale. So it also depends on, it's not only on the field variability, but when Dr. Sharma's team is following all the BMPs to conduct the small plot research, it's like, imagine if we have the ability to put 1 granule of nitrogen exactly where it is supposed to be, right? But we can do that on small plots. But when we are talking about, a 120-acre pivot or 160-acre pivot, we might not be in that place to be that efficient, or we can pinpoint where our fertilizer is, and most of our growers broadcast their nitrogen. So one of the things was that, hey, can we see the same nitrogen use efficiency, small scale versus large scale? Or the other major question was that this is a corn nitrogen rate trial. So after the trial is done for 3 years or 4 years of his time, time that is allocated for the trial, at that time, Dr. Sharma will be ready to present to the PNOC committee, which is our Plant Nutrient Oversight Committee, which will then, based on the data, suggest or will, based on the data and Dr. Sharma's suggestions, will update or not update the corn nitrogen, IFAS corn nitrogen recommendations. So one of the things was that what if we collect actual on-farm data, and if we are able to present that data to the PNOC committee in addendum, in addition to the small plot research, and then let the PNOC committee ponder upon and discuss and then decide what should be the corn nitrogen, the new updated corn nitrogen rate. So I think this is, this on-farm trial is a two-way learning process for basically for us to learn, can we replicate the same precision or nitrogen use efficiency on large scale farms? Or for our growers to learn, is there a window of opportunity for our growers to be more efficient in their nitrogen management in corn? I mean, I know that Dr. Sharma's team is, is following all the BMPs and being very efficient, but as we have all mentioned that on a larger scale, that might not be the case. So this on-farm trials would serve as a learning process for all of us. All right. So these on-farm trials, as suggested by the Corn Advisory Committee, were started in year 2022. In year 2022, we initially began or had accounted for 3 different farms. I'm just showing the data from 2 farms because the third farm had an issue where their main irrigation person left the job, and the new guy who was hired, he messed up the irrigation and fertigation, and so the rates were messed up. So we were not able to collect data from the third farm. And all this data that I'm showing here was again collected by Dr. Lakesh Sharma's team. Dr. Diego Letao is our postdoc, Dr. Sharma's postdoc. So he's the one who did all the hard work. So a huge appreciation for Dr. Sharma's team. So as you can see, on farm 1 was the grower who agreed to apply 4 different nitrogen rates. Now these rates, if you remember one of the slides that I showed where we had all the rates from 0 to 350 in increments of 70 pounds. So this grower had one pivot that he divided into 4 quarters, and each quarter got the rate, 140, the next quarter got 210, the next got 280, the next got 350. And on top of the bars, you can see the yield in bushels per acre. And as you can see that there was, now these trials are not replicated. See, these trials are not replicated trials. So the numbers that we are getting is an average of the yield that we got from that quarter. Now on farm 2, the grower agreed to apply 2 different rates, 280 and 350, and you can see that again the response was all the way up to 350. Whereas if you recall the slides that I showed from our small plot research, the response was all the way to 280. There was no further nitrogen response from 280 to 350. So just by looking at this, we can kind of think that there is a window of opportunity for improving our nitrogen efficiency on the farms. All right. So now moving on to last year, again, we had one farm where the grower agreed to apply 4 different rates, all the way from 140, 210, 280, and 350. The graph that you are seeing here on the screen, the yield data are from 2 different ways that we collected data. On the left-hand side is the combine, so it's the actual data collected while the corn was being harvested. The other is where Dr. Sharma's team measured the plot length and hand harvested corn, and that's the hand harvest data. But in both the data sets, as you can see, there is a response all the way to 350. Now, for this particular grower last year, this is how he applied all these different rates. So after his at plant application, his first split was using banding. So he was being efficient. He was not broadcasting his fertilizer. So he used wide drops for the first 2 lower rates, which is 140 and 210. One split application with wide drops on 2 lower rates. On 2 higher rates, he had the opportunity to use Y drops for 2 splits, and a total of 3 splits were nitrogen was applied in a total of 3 splits for 140, 4 splits for 210, and 5 splits for the highest 2 rates, which is 280 and 350. Now, you might wonder that why these splits were different, because nitrogen was applied in 3 splits for 140 rate because it's so low. 140 rate is so low that, and he wanted to stretch it out all the way to tassel. So that's why he ended up using less splits. Now, farm number 2, for last year's data, for last year's trial, again, the graph is in the same way. One data set is the combine harvest data. The second data set is the hand harvest. And as you can see that when we were just looking at the hand harvest data, the yield for both the rates were, were just a difference of about 2 bushels. But if we are looking at the actual combine harvested data for the entire half of the pivot, because the pivot was split into 2, there's a significant yield difference that we can see. So, I mean, this was really nice to see that the growers are able to see the response all the way to 350, whereas in our small plots we were able to see no response beyond 280. So, but for this grower, he applied nitrogen in 9 splits for the 280 rate and in 12 splits for the 380 rate. And for this grower, there was no Y dropping, there was no banding of nitrogen at the earlier stage. All the nitrogen application was broadcasted. So this is another very nice data that Dr. Sharma's team collected, and this is from small plot research, but I just wanted to make a point that if you guys look at the highest rate on both the sites, which is the 350 rate, and then look at the nitrogen use efficiency, as the rates increase and we move all the way to that highest rate, the nitrogen use efficiency greatly reduces. So, and since we know that this trial has been, maintained and was conducted following all the BMPs, it means that, as we keep on increasing nitrogen rate, that's not going to increase your yield or that nitrogen, basically the use efficiency is greatly reduced. And when we discussed this data with our growers during our corn advisory committee, I mean, they were in agreement. I think that opened up, this data basically opened up a door for really good conversation with our stakeholders, for them to understand that there might be possibilities for them to improve their nitrogen management. And I guess we had really good discussions. So what did we learn so far? I would say that statistical response on small plot research is at about 280 pounds of nitrogen per acre. On-farm response is up to 350 pounds of nitrogen per acre. The only caveat here is that on-farm data is not replicated data, but it's repeated over years. So now we have, if we include this year 2024, so by the end of year 2024, we should have, we will have data from several farms, not replicated, but repeated over years. And so far, what we have seen is that when the on-farm trials, when nitrogen is applied from 280 and moved to about 350 pounds, there is an increase in about 20 to 30 bushels, and that's the response. So that's what we have learned so far. So now the next question is, where can we improve? I think that precision nitrogen fertilizer placement during early growth stages is a key. And some of our growers, as I mentioned, they completely do the broadcast application, and maybe that's the time frame when corn is small, the roots are not developed. Broadcasting your fertilizer might be, basically assisting in reducing your nitrogen use efficiency. So that's the one thing that, we with the corn advisory committee had a really good discussion about. And also one really interesting thing that we had discussed with one of our collaborators was that he would like to increase his number of splits from about 10 to 14 this year, and would try to see if he can get a better response to his nitrogen application. So this whole process was very, it was helping. It was a multi-way communication, both for IFAS faculty and IFAS extension faculty to learn from our crop consultants and our participating growers, as well as for our growers and crop consultants to learn from on-farm data, but to learn more from the small plot research data that Dr. Sharma's team has been collecting. Yes, that's it.